Hello, everyone. Today, we'll be exploring one of the most fundamental and fascinating processes in biology, cell division. All living organisms, from a single-celled bacterium to complex multicellular organisms like humans, rely on the ability to divide and produce new cells. This ensures growth, repair, reproduction, and, in evolutionary terms, genetic continuity across generations. Two main types of cell division occur in eukaryotic cells: mitosis and meiosis. Though they share some similarities in basic mechanisms like DNA replication and chromosome segregation, they are entirely distinct in their purpose, process, and outcome. In this session, we'll systematically compare mitosis and meiosis, feature by feature, focusing on their biological roles, differences in mechanics, genetic implications, and relevance to human health and disease. This will help you not only prepare for NEET and academic exams, but also understand the deeper biological significance of these processes. Let's begin with the purpose of these two fundamental processes. Mitosis serves everyday functions. It's responsible for growth, tissue repair, and asexual reproduction in multicellular organisms. Whenever you heal a wound or grow in height, mitosis is at work. Meiosis, in contrast, has a very specialized role. It occurs only in germ cells to produce gametes, sperm in males, eggs in females, and is essential for sexual reproduction. Importantly, meiosis ensures that the chromosome number is halved, so when fertilization occurs, the diploid state is restored. Mitosis involves a single nuclear division, one round of division that results in two daughter cells. Meiosis is more complex, involving two sequential divisions, meiosis I and meiosis II. The first division separates homologous chromosomes, and the second separates sister chromatids. This double division is crucial because it allows the chromosome number to be halved, and it enables recombination and variation. As a result of its single division, mitosis produces two daughter cells. These cells are exact genetic copies of the original cell. Meiosis, on the other hand, produces four daughter cells. Each of these four cells has half the original chromosome number and a unique genetic composition. This difference is fundamental to their function. Mitotic cells replace lost or damaged cells, while meiotic cells ensure diversity in offspring. The cells produced by mitosis are genetically identical, both to the parent cell and to each other. This is vital for maintaining genetic stability in tissues. In meiosis, however, the daughter cells are genetically diverse. This diversity arises from two key mechanisms: crossing over during prophase I and the independent assortment of chromosomes during metaphase I. This variation is one of the evolutionary advantages of sexual reproduction. In mitosis, the chromosome number remains constant. A diploid, 2N cell gives rise to two diploid cells. For example, a human cell with 46 chromosomes divides to produce two cells, each also with 46. Meiosis reduces the chromosome number by half, from diploid, 2N, to haploid N. So in humans, meiosis takes a 46 chromosome cell and produces four cells with 23 chromosomes each. This is essential to ensure that when gametes fuse during fertilization, the normal diploid number is restored. Mitosis occurs in somatic or body cells. These include skin cells, muscle cells, and any other non-reproductive cells. It allows for cell turnover and growth. Meiosis is restricted to germline cells found in the gonads, testes in males, and ovaries in females. It prepares cells for sexual reproduction by forming gametes. Crossing over is a phenomenon where homologous chromosomes exchange genetic material during prophase I of meiosis. This creates new combinations of genes, increasing genetic diversity. In mitosis, crossing over does not occur. The process aims for fidelity, ensuring the daughter cells are genetically identical to the parent. During meiosis, especially in prophase I, homologous chromosomes, one from each parent, pair up in a process called synapsis. This pairing allows for crossing over. In mitosis, this pairing does not occur. Chromosomes line up individually along the metaphase plate, and no exchange of genetic material happens. Independent assortment refers to how different pairs of chromosomes align and separate independently of each other during metaphase I of meiosis. This results in a wide variety of possible gamete combinations. Mitosis lacks this mechanism because the goal is exact replication, not variation. Therefore, no new combinations are formed during mitotic division. Mitosis is a relatively short and efficient process, typically taking a few hours to complete. It occurs frequently in rapidly dividing cells like those in the skin or intestine. Meiosis takes longer because of its two stages and complex events like synapses, crossing over, and checkpoint regulation. It is tightly controlled and happens only during specific periods, such as puberty in humans. Mitosis ensures that our bodies grow, heal, and maintain themselves by producing identical cells that maintain tissue function. It's also the basis of asexual reproduction in some organisms. Meiosis is significant for evolution and species survival. It introduces genetic variation, which is crucial for adaptation and natural selection. Without meiosis, sexual reproduction and genetic diversity would not exist.